plan is as follows, although it may falter. The plan is as follows. We're going to have a little bit of an introduction by Stan Linnison, who's one of the true leaders in this field. He was there from the start, from the word go, in the single-cell genomics field. And because he hasn't given you a proper lecture, and he's very good at kind of basically exposing all the big questions that the field has, we thought Stan can do 10 or 15 minutes of an overview of what the big questions are, and then we all sit there and squint into the light and can't see you guys raising your hands. We'll see how that goes. And Stefan Lorenz is also going to then very briefly introduce himself, because we haven't seen him in action. And then hopefully you good people have all your wonderful questions ready and raise your hands and everything, and let's see how it goes. Stan.
All right, thanks for inviting me. And yeah, Wolf asked me to try to point to some of the major challenges in the field, and I decided to go back to the beginning, or let's say to my beginning. This will be my personal story, but I started this in, I think, 2007, 2008, thinking about these things. So I went back to one of the first slides that that I made before we had any data or any methods or really anything to show for it, and sort of ask where did we come from? What did we hope to achieve 8 years ago? So here's the slide. The cartoon you see on the right there, of course, I stole it off the internet. It's, I guess, from some computer game. I sort of was imagining that we could one day generate these kind of landscapes that would correspond to cell types. And you would have these islands that corresponded to specific cell types, and maybe, you know, islands that are close to each other would be cell types that are similar to each other, and there would be these bridges between the islands that would show the lineage relationships between cell types. And those are the questions that I asked 8 years ago. The most prominent one up there is what is a cell type? And it's still a mystery, although I think we're now confused at a much higher level, having acquired a lot more data. I wanted to know how many cell types there are. You see I have some suggestions there: 210, 411, or 1,000. I think we can probably exceed that a little bit, but not by orders of magnitude. 210, that's the number from the textbook, Molecular Biology of the Cell at the time. And 411 is still the best number, the most well-documented number from a review by Vicarious et al. That listed all cell types that have names in the human body, and that's 411. Other questions, I think a key question here is about those bridges. So how are cell types related? What are the transitions? How can we go from one cell type to another? For example, naturally during development, cell types are sort of way stations in development where cells are transformed stepwise from more immature cells to progressively more mature and specialized cells. And this is, of course, a branching pattern, and the question is, you know, can we figure this out using single-cell analysis? Yeah, and then I dreamed of this map, of course. In 2009, we had some sort of techniques for doing this. We were not the first or the only ones, but this is where we were in 2009, where we could analyze, I think this is maybe about 200 cells, 200 single cells. Half of them are ES cells and half of them are neuronal cell line. And you can see we managed to make kind of a landscape that has two islands, and we project gene expression onto this landscape. And this actually looks pretty much like our plots look today, except that we have now thousands and thousands of cells, and they are real primary cells from the brain rather than these cell lines. We've taken this relatively straightforward approach to studying these issues, we prefer to simply take cells at random out of the tissue. It's extremely powerful if you don't select cells, but rather take sort of an unbiased sample of all the cells in the tissue, because you get the complete picture of what's in there. And then we just pick cells at random, we generate single-cell RNA sequencing profiles, and then we try to cluster the data. And you've seen many variants of this during today. Last year we published, I think, our first and best real landscape of cell types in a tissue. This was from the cortex and hippocampus. This is the raw data, more or less. This is what you get from such an experiment. And I think this picture is extremely informative, although it takes maybe a little while to get your head around it. Here, this is a heat map of gene expression, of course. So blue is low, red is high. Each column here is a cell type, or sorry, each column is a single cell. Each row is a single gene. And I'm only plotting the genes that have any sort of information in them. So I took away all the housekeeping genes, and I took away all the genes that are not expressed. And then we sorted the matrix, and this reveals this kind of block-like structures that corresponds to major cell types. They've also indicated up there. So this shows one thing that was quite surprising to us when we first saw this, that at least major cell types are characterized more or less by their own sets of genes. So you get these blocks. There are no combinatorial structures on the high level. It's really these very cell type specific blocks. So there's this big rectangle of genes that are specific for oligodendrocytes, for example. So those genes are only in oligodendrocytes. They're not in any of the other cells. And you see the same thing for the other major cell types. Then when you look inside of these blocks, you see a lot more heterogeneity, and there it gets more complicated. So there is this structure, there is this hierarchy to cell types, which I think is an interesting point. Long story short, in this experiment, we found 47 cell types in the cortex and hippocampus. And they correspond to all the cell types that you would expect in the brain, including glia, vascular cells, immune cells, and of course neurons. Maybe the take-home message of this is that when you do analysis in this comprehensive, unbiased manner, you should expect to discover more or less what is previously known, but with a lot more detail. Okay, so there are no new major undiscovered cell types anywhere. That would be extremely surprising. But there are many specific subtypes that are either undiscovered or at least poorly characterized. So since then, we've done a lot more tissues. We're continuing to survey the brain mostly, but we have also done some other tissues, so that by now we have a compendium of about 100 cell types. So now this is a dendrogram of cell types already. There are too many cells here to plot. It's about 25,000 cells. So I've clustered them for you, and then I'm showing the tree of cell types here. And I don't know if you can read this, but in the middle here, there are all these neurons of a lot of different specific types. For example, peptidergic neurons in bluish there. Inhibitory neurons. There's a tremendous heterogeneity among neurons in different parts of the brain. At the top there you have ES cells. They form an outgroup here to this dendrogram. And at the bottom there you have keratinocytes, because we happen to do an epidermis experiment. I'll get back to this in a minute. So, getting to the current challenges and opportunities, I'd like to identify maybe 4 major areas where I think we have made some progress, but where there is an opportunity to make a lot more progress, both conceptually and practically. So the first is understanding cellular identity. So again, what is a cell type? And we've come a long way. We understand now that that cellular identity has structure. I think Oliver Stegel's talk earlier today was good in introducing this, that you have these gene modules that can be activated in cells more or less independently of each other. So cell cycle can be activated in a lot of different cell types, so it's independent of type. And if you go on like that, you will soon realize that most of the functions of a cell can be activated in distinct types of cells, which, if you turn it around, means that cell types are probably not the right way to think about it. Instead, cells express a certain combination of functions, and that's what makes them what they are. So we're going to need a new language for this. There will be some sort of grammar for how you can, what combinations are allowed in cells. Can you activate cell cycle in any type of cell? Probably not. So we need to start talking about this at a higher level. Modules, hierarchy, these kind of things. The second question is much simpler. It's a question of cellular taxonomy. I think we can and should generate complete cellular taxonomies for all of our model species. Right now, the technology is there. It's just to do it. You just need to analyze all the different tissues of an animal and classify them. The third question is about, so a limitation of most, well, I guess all single-cell RNA-seq experiments is that they are static snapshots. But it is possible, and it's been shown to be possible to infer lineage from static snapshots. So the question then is, what are the limitations of our ability to infer lineage? That is to say, for example, differentiation trajectories from a static snapshot. And how should we design experiments to be able to infer lineage? How many cells do we need? If the process that we're studying is very rapid, do we need a very large number of cells? Is it possible to infer from the instantaneous state of a cell what it did express a few hours ago? Are there some remnants inside of the cell that we can use? Let's say half-degraded RNA, something like that, that can tell us what the cell used to be a couple of hours ago. I'll give you some examples of why inferring lineage is challenging. And then the fourth, going to the hospital. Of course, these technologies are extremely powerful, and I think in a couple of areas, these are obvious cases for being introduced into clinic. First, of course, cancer diagnosis and monitoring, including circulating tumor cells, but more generally, taking a tumor and doing a snapshot analysis of all the different cells that are in it, and basing diagnosis on the different specific subclones in the tumor rather than the bulk. So just the same as this has been a revolution in how we study complex tissues in basic science, it is going to revolutionize how we understand tumors in the clinic. We just need to do it. More generally, understanding non-natural cell types. Once we have a cellular taxonomy, sort of a reference atlas of cell types, then we can start mapping diseased tissues onto that atlas. And finally, for regenerative medicine, where we're trying to make tissues, single-cell analysis is going to be an extremely powerful tool to make sure that we are making the right kind of tissues. So let me give you just 3 quick experiments, examples of experiments. Here, I'm showing one case where it's very challenging to infer lineage from a static snapshot. These are oligodendrocytes from the brain. It's about, I guess, 5,000 or so cells. And it's a Tisney plot there on the left. And you can see this rather obvious lineage from the bottom going up and to the right in this sort of banana shape. So you can see the precursors in red there on the bottom left, and then it progresses through newly formed oligodendrocytes all the way to mature oligodendrocytes. Now, any child can draw a line through this and project all the cells to that line, and you can get pseudotime out of this. It's so densely sampled that it's actually a trivial problem in that sense. The real challenge is towards the end of this trajectory, when you see these colors sort of start diverging, where there are really subtle differences that turn out to be region-specific. So when the cells are really mature, they sort of adapt to the local environment. And as we have drawn it here on the right, we don't know how to sort that out. We just draw it as branching out. And we don't know what is really the lineage relationships there. And the other challenge is that this whole trajectory is gradual, as you can see. It's a smooth transition from the beginning to the end. But there is an enormously abrupt transition here at the first stage. So the precursors of all the oligodendrocytes are the OPCs, which are up there in the dashed oval. These are the PGF-alpha positive cells, actually the blue ones up there. So even though we have sampled 5,000 cells, we see no connection between the OPCs and their immediate offspring, the precursors in red down there. So does it mean we need to sample 50,000 cells or 500,000 cells, or what does it mean? Another challenge, of course, if you have branches and incomplete lineages, these are cells from the ventral midbrain, where dopaminergic neurons are born. And here the lineage goes from the orange cells up there, sort of round and to the left, and it branches out in several branches. And I know this because we've analyzed this really exhaustively using all our prior knowledge about this tissue. But the question is, could you infer this from just the data? I think probably not, but maybe with a lot more cells. And then I'll just finish with a possible clinical application. This is for cell replacement for Parkinson's disease. So we collaborated with people who are gearing up to do transplantation, actually here in Cambridge, in Parkinson's disease of currently embryonic tissue from human embryos, but in the future iPS-derived cells that are reprogrammed to dopaminergic neurons. So then we took the human embryo and we figured out what are the cell types in the human embryo, and I'm putting them along this sort of bicycle wheel plot here, the main cell type. So the target cell type is DA over there on the left, dopaminergic neurons, the gray ones. But in the embryo, the thing that you transplant contains all these other cell types. Now if you take iPS cells and you reprogram them in vitro, what you get is the picture here on the right. So you see you're getting sort of dopaminergic neurons, but you're also getting all kinds of other cells, and they're much less well-defined than the ones that you have in vivo. So this shows, I think, in one snapshot, the power of doing single-cell analysis, whereas previously you had this sample and you don't know what it is. You're going to put it in people's brains. Now you can tell exactly what it is that you're putting in people's brains. And then I'll just end with that. Okay. Thanks.
Hello, just a few words to introduce myself. I'm Stefan Lorenz. I work at the Wellcome Trust Sanger Institute, and there I'm running the single-cell genomics core facility. And it's a team of 5 people in total, and we are trying to provide our scientists, like Wolf and Martin, with a standardized single-cell sequencing service. So rather than these high-level academic discussions about cell types and cell identities, we are much more focusing on process optimization and questions like, here's a person that wants to sequence 50,000 cells, how do we make that possible? So for us it's about protocols, standardization and automation, and I hope I'll be able to contribute a slightly different angle to this discussion.
Thank you, Stefan. So I guess the trouble for us is that we can hardly see you guys. So it's going to be a challenge in terms of people raising hands and stuff, but just do fire away. We kind of take any questions. Just put your hand up and we'll start a discussion.
Yes, over here. Thank you, and thank you, Dr. Linson, for that excellent introduction. My question to you and to everyone else is about DNA and whether you believe or are pursuing lineage through DNA studies of mosaicism, because there's a lot of papers on transposon, CNVs, SNVs with very different results, and how much is real and how much should we take it seriously?
I can comment. I think definitely that is going to be a very powerful thing. We are currently limited, I think, mainly by the cost. So to really do it properly, you want to sequence whole genomes from single cells. And that's as expensive as sequencing whole genomes of people. So a project that's of the right size to say something interesting, which would be, I guess, hundreds of cells at least, and in the end you want to do thousands and tens of thousands, it's going to be very expensive at the moment. But once costs of genome sequencing come down, I think that area is going to really start growing.
Maybe you could comment then on the challenge also in terms of the number of new mutations per mitosis. So how many new events do you expect, and how accurate your sequencing needs to be? I think that's another challenge as well. Do you want to say something about that?
I mean, that's why I'm saying that you really want to sequence whole genomes. So it's possible to get around it. In tumors specifically, you can work with CNVs because they tend to accumulate quickly enough that you can see clonal structure, and that's why we've seen many pioneering publications in cancer specifically. But if you want to study normal development, for example, you're limited by the enormous accuracy of cell division. So the cell doesn't make many errors. I think it's on the order of tens of errors per mitosis or something like that.
If that, right.
What?
If that.
Yeah, if that. So, and another limitation is that this is actually counterintuitive, but the body is very well mixed, which means that even if you take cells that are very, very close to each other in some particular part of the brain, two neighboring neurons in the cortex are likely to be as different from each other as one of the neurons and a cell from the heart, for example. That was recently shown by Chris Walsh in a nice paper in Science, which means, again, that you need to sample very large numbers. Most of the mutations will be specific to each particular cell, and that doesn't tell you anything in terms of lineage. It can tell you other things, but not for lineage. So many cells will be needed.
Very nice talk. Thank you very much. I'm a biologist from Milan. And my question regards the microRNA repertoire. So they are very specific. So did you address this on all of these cell types? Yes? And
yeah, no.
Can you combine then this with gene expression?
Yeah, so there are limitations. We use oligo-T-primed cDNA synthesis, which means that we get mostly regular mRNAs, protein-coding mRNAs. We do get some non-coding RNAs if they are polyadenylated, and we do get the occasional microRNA precursor, which also is polyadenylated, but we don't get microRNAs systematically, and certainly not the processed microRNAs. So I don't think that there are currently any methods that will allow you to get a regular transcriptome and a microRNA transcriptome at the same time. But you can get certainly microRNAs independently of the transcriptome from single cells.
Yeah, you just mentioned it. So there was a poster in Utrecht at the single cell meeting where someone demonstrated microRNA sequencing on single cells, but the way it's done currently, it's not compatible with messenger RNA sequencing. So I guess that's a challenge for the method developers in the community to come up with a method that ideally captures all RNA species. So not only mRNA and microRNAs, but all the long non-coding ones as well. But I'm sure in a year's time we'll have a method.
Yes, I agree. For long non-coding RNAs, the challenge is that there are methods to get the non-polyadenylated fraction but they tend to be much more expensive per cell because you are sequencing a much larger pool of RNA. You need to go through all the ribosomal RNA. It's difficult to deplete ribosomal RNA properly from single cells. So it's a bit of a trade-off as well, and I suspect that the reason we haven't seen more efforts to really capture all of the RNA is that once you think about it, you have to pay a heavy price for doing that. You will do much fewer cells. But one day it will all be very cheap, so we'll have it.
For this single-cell genomic sequencing, what is the error rate from your experience in the data that you've seen in terms of the mutations induced by the polymerase? And second separate question is, what is the best control if you have a let's say, cancer single-cell sequencing? What is the best control for the individual's normal sequence?
The error rate for Phi-29 is published as 10 to the minus 10. 10 to the minus 7. So it's relatively low. It's 1 in 10 million. However, there's a specific error profile for the